By the end of this video, you will have improved your exam technique, understood the main concepts of the immune system.
Alright guys, so this is the first question. Let's pause the video now and try to solve it. So this is a 3-mark question, and the first thing I need you to do is read the question carefully and highlight the keywords in the question. So the first 3 lines of the question, I can see that it is not actually the question, it is just a summary explaining that the human body has several barriers. So you want to read it carefully from here. So this is the bulk of the question. Explain why the presence of microorganisms, you want to highlight microorganisms, on the skin and in the gut helps to prevent pathogenic organisms. So prevent pathogenic organisms. So what we're trying to explain, or what they want us to explain, is how the normal presence of microorganisms in the skin and in the gut helps prevent pathogenic organisms. So the relationship between normal organisms, or bacteria that are normally present on our skin and on our gut, how do they help us fight harmful bacteria? So for those who are not aware of the concept, we have a normal flora or a gut flora. We call it a gut flora. This gut flora or skin flora, what I mean by flora is a normal layer of bacteria that lives permanently in our gut and in our skin. Not just humans, it's on all mammals, right? So this flora is composed of a lot of bacteria that are not harmful, that are not trying to harm our bodies. They're not trying to invade our bodies. They just live in the gut and in the skin, and they feed on scraps of nutrients that you give them, or that you feed on, or that fall on your skin. So this gut flora, how does it stop harmful organisms? So let's read the answer. Let's read how I answered this question. These organisms are non-harmful microorganisms in the skin and in the gut. So I'm talking about these microorganisms on the skin. The gut and skin flora, now I named them. So you have to name them. Show the examiner that you know that these microorganisms are called the gut and skin flora, are well adapted to the conditions. So this term, well adapted to the conditions, this is the first point. That this is the advantage that the gut flora has over the harmful bacteria that is coming to invade the body. The harmful bacteria is not adapted. It's a new environment to it. While the gut flora has been there for years and years and is adapted to this environment. So this is the first point. The guts and skin flora are well adapted to the conditions in the gut and the skin. Therefore, when harmful pathogens enter the gut or the skin surface and try to compete, this is the next keyword, competition. This is what happens. So harmful bacteria comes to the gut or comes to the skin and tries to compete with the bacteria that is already present for nutrients, for space, for temperature, for different resources. Who wins? When the harmful bacteria comes, who wins? The normal flora wins because it is well adapted. It's playing at home, basically. So it's its own home, so it's adapted more. So when it competes with the flora, the gut and skin flora outcompetes them. So when it outcompetes them, the harmful bacteria finds itself without nutrients, without a right place to live, so it eventually dies, and they eventually die. Additionally, this is one point. This is one way in which the normal flora prevents harmful bacteria or harmful pathogens from entering through the gut and the skin. The second way is that the gut flora specifically, bacteria in the gut, secrete chemicals to kill harmful pathogens. So they act like an antibiotic or an antiseptic. Bacteria are killing other bacteria which are harmful. This last sentence is a summary of what happens. This is in the question, so I won't get a mark for it. So you get a mark for explaining how the gut flora is well adapted. You get a mark for explaining the concept of competition between them and mentioning who is going to win, so out-competes. And you get a mark for explaining how gut bacteria secrete chemicals to kill the bacteria of the harmful pathogens. So this word pathogens here, let's talk about this word for a bit. This is a very good word to use in your exams because this word means a bacteria or a virus or a fungi, any organism that causes disease, any organism that targets the body and tries to enter to cause disease. So an organism that causes disease is called a pathogen. Let's go to the next question. Pause the video right now and try to solve the question, 4-mark question. You will have 5 minutes to answer this question. Read the question carefully and answer it. All right, so this is a very important question about inflammation. It's quite a direct question as well. They're asking you, explain how changes in the blood vessel, so we want to be talking about purely about the blood vessels, changes in the blood vessels and how they cause redness and swelling. So this depends on your understanding of how redness and swelling happens when an actual cut or an infection or a bump happens to the body of a human. So when you get a bump or a fall, or you bump into something, or you get cut with a knife, this area becomes red and becomes swollen. Why? This is due to a concept called inflammation. And this inflammation is present so that your body defends itself from pathogens that are trying to come in through this cut or through this bump. All right? So inflammation is a concept in which the body defends itself. It's a non-specific response. It happens no matter the pathogen. If you get infected with a virus, if you get infected with a bacteria, with a fungi, with anything, inflammation will always happen. As soon as a cut is made, inflammation will happen. So we call it a non-specific response. It does not care which specific pathogen came. All right? So now let's explain the changes in the blood vessel. So the first thing that happens when a blood vessel is cut, or when a blood vessel, or when the skin is compromised, is that white blood cells surround this place. All right? So the white blood cells surround this place and they detect that there is an intruder in the body. So someone came in from outside. So in response to that, they release a chemical called histamine. And this is very, very important in the process of inflammation. They release a chemical called histamine. And this histamine, what does it cause? It causes the blood vessels surrounding this region to dilate. So if they were, this is vasodilation. If the blood vessel was this thick, it causes the blood vessel to become this thick. Therefore, more blood is going to flow into this region. So it's like asking for more blood. The histamine is asking for more blood to come here. Why is it asking for more blood? So that more and more white blood cells come to the region. More and more soldiers come to this region to fight off the infection. All right? So histamine is released. Histamine causes the blood vessel to dilate, and this is called vasodilation. Vaso means blood vessel. Dilation means to become bigger or thicker. So this is vasodilation. Now what happens when vasodilation happens? You have a lot of blood causing pressure on the capillary wall. Let's say this is a capillary. So the capillary wall has a lot of blood pressure because there's a lot of blood flowing through it. This causes the capillary wall to become leaky. We use this word leaky. This is also a scientific word. You can use it in the exam. The capillary wall becomes leaky. So it begins to leak the liquid part of blood. Okay, so this liquid can actually leak through the holes in the capillary and get out into the surrounding tissue. So this area gets filled with fluid, and what leaks from the blood is called the plasma. Plasma is the liquid part of the blood. So it's not the red blood cells that leak, it's not the other parts of the blood that leak, the proteins. They don't leak out, they're too big to leak out through these holes. What leaks is the water part, and everything dissolved in the water, and with it the white blood cells start leaking as well. So here you have a lot of liquid with a lot of white blood cells inside of it. All right, so this liquid is going to cause the swelling. The swelling that you get when you cut yourself is caused by a lot of liquid leaking from blood in that region, and it's accumulating under the skin. So this is what's going to cause the swelling. Let's look at our answer here and highlight the important keywords that you have to have in your answer. See, notice how I organized my answer step by step. It's like you imagining the process in your mind and listing it step by step. The first thing that happens, when a pathogen enters the body, white blood cells release histamine. So the release of histamine is the first step. It's going to carry 1 mark. Around the site of entry, so the site of entry of what? The pathogen. So histamine is being released at the site of entry. What does histamine do? This is the second step. What does histamine do? Histamine causes vasodilation. All right? So I'm using the keyword. Instead of saying it causes the blood vessels to be thicker, we are at A level right now, we have to use the keyword vasodilation. All right? Vasodilation of blood. Blood vessels, and this causes more blood to flow to the area, causing redness. So more blood flows to the area. More blood flows to the area. More blood flows to the area. This is also a very important point. What happens after vasodilation? How is vasodilation useful? More blood flows to the area, and this causes redness. So I linked everything that happened to the redness. So why does the redness happen? Because there's a lot of blood in this area and not a lot of blood in this area. So this area is going to be redder than the other areas of the body. So now I answered half the question. How does changes in the blood vessel causes redness? Now I need to explain swelling. So the increase in the amount of blood causes the capillaries to be leaky. You can use the word to be more permeable as well in this instead of this keyword. So leaky is accepted and permeable is also accepted. Both are scientific words. Allowing plasma to leak into the surrounding tissues. So you have to mention that it is plasma that leaks. It is not blood that leaks. So not the whole of blood leaks. Red blood cells do not leak. Proteins do not leak in blood. Only the liquid part called plasma leaks through the capillaries, causing swelling. So now I link this with the swelling, and this will give me the 4 marks for this question. I always want you to be organized. I want you to be organized, and when you have a process like this, you list it in a stepwise manner. And you have to brainstorm. Before you answer the question, you have to brainstorm. What are the points and in what order I'm going to list them? I don't want you to start writing, and then as you write, you think of things to write. This causes a disorganized answer. You don't want to do that. You want to be to the point, and you want to be organized. The more organized you are, the less points you miss, and the more marks you get. So you get a 4 out of 4 because you're organized. Next question, also about immunity. You have 5 minutes to answer this question. Pause the video now. All right, so for this question, it is a direct question asking you to explain the role of T cells. It doesn't matter which pathogen they're talking about in the question. What matters is explain the role of T cells in immunity. Here in this question, they're talking about the Ebola virus, but as long as you know what T cells do and step by step their role in immunity, you will be able to answer this question. Now, this is a 3-mark question, so we don't need to go into so much detail about antigens and about whatnot. We just have to be specific in the important points that T cells do to link between phagocytosis and antibody production. So T cells are the link between these two processes. Now I have an answer in red above the question, and this is my brainstorming process. So when you brainstorm, I told you, whenever you have a process that involves a lot of steps, you brainstorm first, you start thinking about the points you're going to mention in order. If you have a rough sheet in the exam, you can make a mental plan or a mental map, just like this one. I mentioned 6 points that have to be inside this answer. All right, so these 6 points, 3 of them are going to give me the mark. I don't know which ones, but I know that this is everything that T cells do. All right, so look at these. Take your time to read these 6 points and see how I arrange them in order. Now it makes my answer easy. So it may take me like 3 minutes to write these 6 points, but once I have them, I can write this answer within 1 minute or 2 minutes because I already have my mental plan. All right, so while this method might seem to you that it takes a lot of time, no, it takes a lot of time to come up with these steps, which is the answer, but I just need to fix it a little bit to make it a paragraph. All right, so let's look at the answer and see how many keywords we can find, which have to be in this answer. So I started my answer with T cells are of 3 types: T helper cells, T killer cells, and T memory cells. So they're talking about T cells in general. They didn't talk about only T helper or talk about only T memory. They're talking about T cells in general. So I started by mentioning all the T cells, and then I begin with the first T cell that begins the role of T cells. So the role of T cells begins with the T helper cell, going and meeting the antigen presenting cell. This is the first step of specific response, of the specific response. The non-specific response ends at phagocytosis, and then the phagocyte presenting its antigen on its surface, so becoming an antigen presenting cell, an APC. This APC is going to be met by the T cell. So this is the first step where the T cell is involved. T helper cells begin by binding to the APC. So this is my keyword here, binding to the APC. I didn't say meets the APC. I didn't say, so binding. You can say binding to the antigen on the APC. So binding is the word used to describe this meeting of receptor with the antigen. APC, you don't need to say antigen presenting cell. You can use the abbreviation, APC. And detecting the antigen on its surface. Once the antigen is detected, the T helper cell divides. You can use divides, you can use proliferates. All right? So as long as you mention that, more T cells begin forming. Multiplies, you can use multiplies further into many T helper cells and memory T cells. So you have to mention memory T cells. When it binds, it detects the antigen, and then it makes a lot of T helper cells and a lot of memory T cells that are specific to this antigen, complementary to this antigen. The T helper cells activate. This is another keyword. So the T cells actually activate the B cells. This is the main function of T helper cells. They detect the antigen so that they can go to the B cells and activate them. So the T helper cells activate specific B cells to start dividing and form plasma cells. So when B cells get activated, they start dividing as well. And what gets formed? Plasma cells. So plasma cells are special cells that make antibodies. So they are also B cells. They are a part of B cells, a part of B lymphocytes. They produce antibodies. This is also a common multiple choice question. Which type of cell produces antibodies? Plasma cells. Plasma cells can secrete antibodies. Secrete, meaning to release it out into the blood, into the body, to release antibodies, to make them and release them, which are specific against Ebola virus. Here I included the virus that the question mentions. whatever the virus, you can include it. The memory T cells will remain in the body. Now I'm mentioning the function of memory T cells. Why are memory T cells present? What are they good for? The memory T cells will remain in the body for a long time in case the body gets infected with the same pathogen again. You can either say same pathogen again, or you can say reinfection. Why do we have memory T cells? So that when the same pathogen infects the cell again, or infects the body again, the response becomes faster. I don't need T helper cells to go and activate B cells again. No, I already have memory T cells that are already ready and notice this antigen, recognize this antigen. So this will cut the response short. So for example, you get a common cold or an influenza, and you get a cold for 2 weeks until your body gets rid of it, or your immune system gets rid of it. You have symptoms for 2 weeks. In comparison, you get infected with the same influenza virus again after a few months. The cold or the symptoms happen for maybe 3 or 4 days in the second time. This is because when the influenza virus infected your body the second time, you already had memory T cells stored in your blood that can immediately react and activate antibodies against this virus because it can recognize it straight away. It's not the first time it has seen it.
Feel free to check out my other exam technique videos and see you next time.